So our body receives sound, light, and smell signals. Signaling is very important because without it we would die. So after the cell detects a signal, it will amplify it, and once our body integrates the signal, we will respond accordingly. So primary messengers are going to tell you sound, light, smell, basically the five senses. And there are special receptors that receive these primary messengers in the body. So there are two types of receptors based on the cellular location. The membrane-bound receptors that are present on the plasma membrane, like this one right here, this is going to be extracellular, while the intracellular receptors, like this steroidal hormone receptor right here, is going to be found in the cytosol. So steroid hormones are very lipophilic. Basically, they contain a lot of hydrocarbons, so they can translocate through the plasma membrane very easily without a transport protein. So once the steroid hormone is in the cytosol, they're going to bind to their specific receptor. This is going to be the estroidal receptor. These estroidal receptors act as transcription factors because once they bind with the hormone, the receptor is going to go inside the nucleus, and they go inside the nucleus either through a transporter or endocytosis. So the reason why the receptor protein is found in the cytosol and not in the nucleus is because we don't want the hormone to travel through two membranes to get to the receptor protein. It's only going to travel through one membrane. So many transcription factors stay in the inactive state, and they are usually dimers or trimers, basically more than one protein put together. And they are activated by the ligand. They become separated, and the active part will go inside the nucleus. So, once again, inside the nucleus, the active part of the receptor with the steroid hormone is going to bind to DNA, which regulates gene expression. So the gene is either going to be repressed or activated, and then when the gene is activated, the mRNA is going to be synthesized, which travels outside of the nucleus to lead to protein synthesis. So most of the receptors that our body deals with are the plasma membrane bound receptors, which can be split into two categories. The 7-TMR right here, it's going to stand for 7 transmembrane receptor, and then the tyrosine kinase receptor. So just like the name, the 7-TMR is going to have 7 different transmembrane domains. So 1, 2, 3, 4, 5, 6, 7, and they're all going to be connected by loops. The extracellular beginning of the 7-TMR is the N-terminal. It's called the N-terminal because of the amino group attached, while the intracellular part is going to be the C-terminal. So the hormone or the ligand is going to bind to the receptor from the outside, and keep in mind that a ligand can either be an agonist, which activates the receptor, or it can be an antagonist, which prevents activation. So right here we see the C3 loop located on the inside of the membrane. The C3 loop is going to interact with G proteins. Oh, and also another name for the 7-TMR is G protein coupled receptors, or GPCR. So here's an example of the tyrosine kinase receptor. So as you can see, this picture depicts 4 different proteins, 1, 2, 3, and 4. It has 2 alpha subunits on the outside, and then 2 beta subunits that extend through the membrane. So one receptor contains 1 alpha subunit and then 1 beta subunit. So basically one receptor is just this half right here. And then the 2 the binding of insulin brings the two receptors together, and this is going to be called dimer formation. So once the dimer forms, the two beta subunits are going to phosphorylate each other, and phosphorylation makes the entire receptor active. So it's going to go from the hormone binding to dimerization, which is the subunits getting closer together, to autophosphorylation. So remember that tyrosine kinase receptors are transmembrane proteins, while the GPCR, this thing right here, is a 7 transmembrane receptor. And the tyrosine kinase receptors, like this insulin hormone receptor, are different from GPCRs because the conformational change that is seen in the insulin hormone receptor allows them to be phosphorylated and then become active. So this picture shows what happens after the hormone is already bound to the GPCR, causing the GPCR to go through a conformational change, making the receptor active. So the active receptor is now going to activate a G protein, seen right here. The G protein is a trimer. It has a alpha, gamma, and a beta region. So the alpha subunit is going to be bound to GDP right here in its inactive state. So activation is going to lead to the exchange of GDP into GTP. So once GTP binds to the alpha unit, it's going to dissociate from the GPCR and the beta gamma subunits, as we can see right here. So the active form of the G protein is going to be the alpha subunit attached to GTP. So once the active G protein does its job, it's going to need to be inactivated. The G protein is inactivated by an enzyme called GTPase, which converts GTP back into GDP, inactivating the G protein. So there are a lot of enzymes that can be activated or inhibited by G proteins, like adenylyl cyclase, guanylyl cyclase, and phospholipase. The reaction between the enzymes and the G proteins is going to produce secondary messengers. The secondary messengers are cyclic AMP, cyclic GMP, and then IP3 and DAG. So this table, we don't really need to memorize it, but it just shows how diverse a class of molecules can be, and it also shows that the receptor types outnumber the hormones. So right here we have a beta receptor, which is stimulated by epinephrine. Once epinephrine binds to the receptor right here, it's going to be, the receptor is stimulated and it activates the GS protein, which is just another class of G proteins. So the GS protein is going to go and activate adenylyl cyclase and produce a second messenger, cyclic AMP. So CAMP performs a lot of different functions, like causing vasodilation, heart contraction, and the production of urine. In the brain, increased cyclic AMP gives you a fresher feeling. So anyways, cyclic AMP is going to then bind to PKA, or protein kinase A, and it's going to lead to glycogen breakdown, as seen over here. In another mechanism, the active PKA that's bound to cyclic AMP can also get inside the nucleus and serve as a transcription factor, and this is going to regulate gene expression. So nitric oxide can also work as a secondary messenger. Some receptors, after they are activated by a hormone, can increase nitric oxide. Nitric oxide is produced by all kinds of cells in the body, and it used to be called EDRF. And also nitric oxide basically causes vasodilation. So here we see a blood vessel. Blood vessels are made up of smooth muscles, but inside there is a unilayer of endothelial cells which produce a lot of nitric oxide. So nitric oxide is going to leak out of the endothelial cells through simple diffusion and then go into the smooth muscle right here. And then once that happens, nitric oxide is going to act as a secondary messenger and activate a protein in the smooth muscles called S-guanylate cyclase. Then it's going to go on to produce cyclic GMP. So phosphodiesterases degrade cyclic AMP into and cyclic GMP into 5' AMP, and then cyclic GMP into 5' GMP. This basically means that the phosphodiesterase will reduce the effect of the secondary messengers, CAMP and cyclic GMP. So Viagra is going to inhibit the enzyme phosphodiesterase, which in turn keeps the concentration of cyclic GMP elevated, which causes vasodilation. So again, Viagra inhibits this enzyme, and when this enzyme is inhibited, high amounts of cyclic GMP are going to be found, which is going to cause vasodilation. So methylxanthines are caffeine and theophylline. They also lower the activity of all isoforms of phosphodiesterases, which basically produces a lower effect similar to that of Viagra. Then lastly, calcium is a secondary messenger, and it is also a cofactor for calmodulin. Calmodulin will either stimulate MLCK to contract the muscle, or it's going to activate phosphodiesterase, which will bring down the concentration of cyclic AMP and cyclic GMP, which in turn is going to cause the muscle to relax. Alright, that's all I have for you guys. See you guys next time.